So another paper came out on IgG4s in mRNA vaccinated individuals. This one came from New Orleans, so US. And what's super interesting about this particular publication is that the type of work that the authors involved did allows us to start making comparisons with theories of Dr. Geert vanden Bossche, someone who I've been following very meticulously, very closely. And the reason why is because, as far as I'm concerned, he's the only scientist who is currently predicting pandemic outcomes, and he's also the only one that, as far as I know, who accurately predicted that the virus is going to undergo certain evolution and allow immune escape from the antibodies that we basically attempted to destroy the virus with. Okay, so and that's basically how we ended up with Omicron. And then after Omicron, we all know what the history is. Everyone was starting to get infected. So very, very interesting information, very interesting work. So we're going to tie that together, and we're going to also then update this information with what Dr. Bossche thinks about IgG4s in this context as well. All right, my name is Dr. Mikola Rashik of Neurogenomics. Let's get going on this topic. It's convoluted, so be patient with me. So first of all, let's summarize the publication itself. And the authors confirm basically, hey, these IgG4 antibodies start showing up in mRNA vaccinated sometime soon after second vaccination. So we've seen now multiple publications showing this. And after third mRNA vaccination, provided that these individuals never had infection first prior to vaccination, after third mRNA vaccination, basically everyone appears to have these IgG4 antibodies. Now, I'll get into the IgG4 antibodies as to why that's of significance later on, so don't worry about it for the moment. But there's couple things that was remarkable about their work. Well, number one, so what I just told you confirms what we've seen already in multiple publications before. So one thing that they did is, number one, they showed that these IgG4 antibodies, most of these papers, when they look for the IgG4 antibodies, they look for their ability to interact with the spike protein receptor binding domain. So that's the part of the spike protein that interacts with the receptors in our body, ACE2 receptors. So the part of the spike protein at the very tip of the spike protein, that's called receptor binding domain. And that's how we normally, or these scientists who've been investigating IgG4s, they've been looking at how well these IgG4s, or the antibodies that they're looking at in general, all subclasses of IgG, they're looking at their ability to bind to that receptor binding domain. These authors did that, but they also looked at general binding to the spike protein as well. The reason why this is interesting is because when they looked at the IgG4 binding to the receptor binding domain, they looked at people who were vaccinated twice and then had three shots, and they looked at time points, and they looked about just under 1 month after each shot, and then 6 months after the shot. So you could tell after the third injection, mRNA injection, this was Pfizer injection, most of these studies are on Pfizer, they were able to see that there is a drop in IgG4 antibodies against receptor binding domain. Now, technically that's good news, because these IgG4 antibodies are dropping in their amount. That's good, because that's potentially one worry, is that we're increasing amount of unusual IgG4 antibodies in our body, and we don't want that because of their potential pathogenic roles. And we'll get into that, hopefully, if I don't forget. So, but when they looked at, so that's seemingly good news, and I talked about that in one of the previous videos, that it's been observed before. So all is great. But then when they looked at the IgG4s against spike protein, targeting spike protein anywhere, they didn't see a drop. So what does that mean? Well, first of all, that all the levels of IgG4 against spike protein, they seem to be staying constant. But what does it mean if the IgG4 levels are dropping against receptor binding domain? It means that there's like some sort of a shift going on where they're no longer recognizing that area of the spike protein, and there's a shift to recognition somewhere else, which is, by the way, one of the fundamental theories of Dr. Geert van Bossche. Now, very quickly about Geert van Bossche, he somewhat became infamous, and he's totally ignored by science, like absolutely ignored by science. I study his science because of his explanations, and I really enjoy studying his explanations with regards to innate immunity, right? So the reason why he's infamous is because he's predicting that the virus will undergo another type of evolution where it will become more pathogenic, and it's not gonna be a happy ending, right? So that's kind of like why he's not popular. It's just not a theory that you want to be studying and hearing, because he predicts something really bad is coming. And basically, the essential part of his theory is that you have to have a switch of antibodies from targeting the dominant areas of the spike protein to less dominant areas of the spike protein, such as no longer targeting receptor binding domain as much and starting to target other areas of the spike protein. So this observation was interesting in its own right, in terms of these authors showing, hey, these antibodies are no longer IgG4 antibodies are no longer binding receptor binding domain as well, but they're still binding spike protein at the same level. So some shifting is going on there. Another interesting thing that these authors did, though, is that they're pretty much the first one, as far as I'm concerned, that basically called that as a consequence, the longevity of these vaccines is not good. Basically, what's happening that is this repeated immunization is causing problems, and they're basically vaccines work well, but not for a long period of time at all. And another thing they did is they looked at a variety of different variants and how immunization was being able to deal with different variants. And as people were being vaccinated, they dealt well with the original variants prior to the Omicron emergence. But once Omicron came around, further immunization didn't do very well. It could still do a little bit of dealing in terms of binding the spike protein. Neutralization assays showed that they could block to some degree the Omicrons as well. But in reality, it just was nowhere near as good as what they were seeing with the earlier variants. So that's the conclusion. And again, what they're saying is something, they're reconfirming as to what other authors are saying. Why are these IgG4 antibodies showing up? Is because, look, there's B cells who've already been triggered to start producing antibodies. So they refer to them basically that they've already encountered antigen and they've been induced to produce certain antibodies, but then they re-encountered the antigen again and again. And as a consequence, they were induced to start switching the type of antibodies they're producing. This is called class switch recombination, I believe, and they were induced to start switching from the dominant neutralizing antibodies to IgG4 antibodies. And they also made a comment that, look, these antibodies, IgG4, because they do not participate in clearance of what they target as what you would normally expect from normal antibodies, what they're suggesting is that, look, we need to be investigating this is because they might allow persistent presence of a virus in mRNA vaccinated individuals that might have these IgG4 antibodies. Hey, Let's see if I can show this. Like what I found, I think it's a statue of Nikolai Copernicus, but the plaque is missing. But there's a date, 1974, commemorating 500th anniversary of the dude's birthday. And well, he's got the same name as me. So I thought that was neat that I found it here in the park. Anyway, let's go on. So now, what's super interesting about this author's work is that they've done some one-of-a-kind study with the same patients that were involved in the study I just told you. So same patients, they've already published the study earlier in the year, and what they did is, again, as far as I understand, it's the first kind of study of its kind where they looked what do these patients' antibodies bind to after serial vaccination. So these individuals, as I said, had 3 vaccinations. They basically wanted to see, hey, what happens with these antibodies as these individuals keep getting vaccinated? Do these antibodies start targeting anything else, or do you compounding what they are designed to attack, the antibodies? So they wanted to investigate this. And this is where we're going to start connecting to Dr. Geert vanden Bossche, and I'll explain that in a moment. So then going back to that study, this earlier study, so what they did is they had samples, blood samples of these individuals after about a month after each vaccination. So obviously dose 1, 2, and 3, because all of these individuals, they had data only up to 3 doses. And they did really, really interesting analysis. What they did is they took a virus, and these viruses were made to express fragments of spike protein on their surfaces. And basically they took the antibodies from the blood of these patients and they looked, what do these antibodies bind to? Once the antibodies bound to the virus with specific fragment on its surface, fragment of the spike protein on its surface, then they were able to decode this genetic sequence of the virus inside, and they could determine exactly, hey, this is what the antibody was targeting. By the way, the target of antibodies, that's called epitopes. Now there's some limitation here, is because remember, proteins are made of amino acids, and so amino acids are building blocks of proteins, basically a bunch of different chemicals, and you can string those chemicals together in a row and you make a chain of these. But this chain of chemicals, of these amino acids, they can start making 3-dimensional shapes, and that's how you in the end up with spike protein. It's a single chain of amino acids that eventually starts intertwining, and voila, you end up with 3D shape of a spectacular spike protein. So what they did is they took fragments, and fragment of a protein is called a peptide, and they basically, those are the peptides that were shown on the surface of these viruses. So that's a limitation, because number one, sometimes these peptides, they're just linear, meaning they don't actually produce even any 3-dimensional shape, versus spike protein has obviously very complicated 3-dimensional shape. There's a lot of nooks and grooves and so on, lots of space where chemicals can interact with the spike protein and so on. So that's a limitation. Now some of these peptides can be long enough that they can start adopting 3-dimensional shape, but sometimes you can have amino acids of the protein come from very distant location in the chain, but they come together in a 3-dimensional space. So for example, amino acid that's the first one in chain can end up being close to amino acid number 100 in a chain. They're in proximity in space, even though they actually have 100 amino acids in between, type of thing. So this type of epitopes, called discontinuous epitopes, which still would form 3-dimensional shapes, this method cannot analyze at all. So it only looks at fragments, linear fragments of the spike protein. They were able to do this for at least 10 different coronaviruses. So they did obviously SARS-CoV-2, the virus that is obviously still plaguing us right now. They did the first one, SARS-CoV-1 from 20 years ago. They also did MERS. That's the deadliest coronavirus, as far as I know, that we ever had to deal with. And they also did the common cold viruses. These are also coronaviruses. And then the rest was bat coronaviruses. And they basically, all of these viruses, they were able to get something like about 56 fragments, those linear peptide fragments per each spike protein of all of these different viruses. And they basically tested to see what happens to the antibodies of these individuals once they were immunized. So once they took Pfizer shot 1, 2nd, 3rd. All right, so that's what they analyzed. That's what we're going to be looking at. And what did they look at in this study? They looked at 3 things. They looked whether the number of targets by the antibodies increased. So they call that increasing breadth of targeting. So that's what they looked at. They looked at how much of each target was being targeted by the whole group. So they looked at the percentage of targeting. And then finally, they looked at the magnitude of targeting. So basically, how powerfully each fragment was being recognized by the antibodies. And this is where it gets really interesting. Now, what they were able to observe is that, in a nutshell, repeated vaccination increased the breadth of targeting, meaning as these individuals keep getting more vaccines, basically after the second one, especially after the second shot, the number of targets increased quite dramatically. So basically more areas were being targeted by the antibodies produced as a consequence of being vaccinated. All right, so there's that. And the magnitude also increased, meaning what the antibodies were targeting, they appear to be targeting even stronger. Okay, so there's that. And then they basically also looked at how frequently was that observed in different individuals. So different things were being observed in different individuals. Some people had really strong increased breadth of targeting, meaning number of targets. Some were the same. Some even dropped their number of targets. But overall, there was, amongst this number of patients, overall, there was an observed increase, okay, of number of targets and the magnitude. But what's really interesting is that, number one, first of all, if you look at these figures, even prior to any vaccines. So when these people were naive, they were not previously infected by SARS-CoV-2, as far as these individuals know. Oh, too much wind. Let's get out of here. All of them actually show certain level of targeting right away with their antibodies in the blood against SARS-CoV-2. Very interesting. What does that mean? It means that just exposure to most likely common colds had the capacity to give us antibodies to target the spike protein anyway. And they saw this cross-reactivity across basically all viruses that they tested, because they were able to show, for example, that of about over 400 different peptides across 10 different viruses, at least 200 of them were being bound by the antibodies of all these different individuals. So clearly, spike proteins of all of these different viruses are being targeted by the antibodies. So that means there's a lot of cross-reactivity. That means there's a lot of binding to parts of spike protein that are conserved between species, between different versions of this virus, right? Whether it's common cold, whether it comes from bat coronavirus, or whether we're talking about SARS-CoV-1 or 2. So there's fragments of spike protein that are conserved in between these spike proteins and these antibodies are targeting. On top of that, what they were also seeing is that as people were being vaccinated, there was definitely observed shift to at least 5 new areas of the spike protein, and they looked at basically across the entire spike protein. So clearly there was a shift from some dominant areas to new areas, and these new areas were away from receptor binding domain. Some of them overlapped a little bit the receptor binding domain, but the best neutralizing antibodies are known to be able to target the receptor binding domain. The shift was appearing to be away from receptor binding domain. So completely different areas of the spike protein, and they actually showed which areas of the spike protein were being targeted. So why is this important? Well, first of all, there's some surprises. They didn't see receptor binding domain being targeted much at all in their study, and that's unusual because that's the one area that you expect to be targeted a lot. The reason why they think they weren't able to see that is because of the fact that very likely those linear peptides, and receptor binding domain creates three-dimensional shapes from very different corners of the protein. So we're talking about amino acids that are along the chain very far apart from each other can form the receptor binding domain. So that's why they're not seeing. But nevertheless, very clear observation that there are shifts towards how these antibodies, what they're targeting. So they're starting to target, as these individuals keep getting vaccinated, there was a shift towards recognizing new areas of spike protein. So the authors thought, hey, this is great. Oh, and by the way, this also coincided with increased neutralization of the virus, of pseudovirus. That also seems seemingly good. All of this, these authors are saying, hey, look, it's great. We're seeing increased new targets, we're seeing increased magnitude, we're seeing completely new areas of the spike protein that are being targeted, that are being cross-reactive, meaning it can recognize many different variants, many different spike proteins, all great. And that's how they concluded the paper. Here's where it finally gets interesting, because we're now gonna bring Dr. Geert vanden Bossche into this. And Dr. Geert vanden Bossche basically says that in order for the immune escape to occur, meaning the evolution of the virus took place so that it could start dodging the antibodies that we're throwing at it, the virus had to evolve in such a way so that steric immune refocusing took place. And that's what helped the virus basically evolve. Now, what is this steric immune refocusing? In a nutshell, it means that the antibodies that we were producing as a process of vaccination started to target new areas of the spike protein that were less and less dominant and more subdominant, different areas of the spike protein that normally should not be targeted. And these subdominant areas, basically, they end up being weaker antibodies targeting more conserved areas. Exactly what these guys were seeing in this publication. So basically their results confirm theories of Dr. Geert vanden Bossche. And Dr. Geert vanden Bossche is saying, look, yes, these new antibodies being produced, they're being produced is because when the virus was mutating so that the original antibodies were no longer recognizing the virus as well as originally, well, they were not able to neutralize the virus, but they were able to still block the virus. So that means our body was forced to start producing new targets, new antibodies against new areas of the spike protein, because there was like a blockage of being able to retarget the dominant areas we wanted. Why? Because they mutated a bit, right? So that's the steric immune refocusing, exactly what these guys were seeing. And because of steric immune refocusing, these new antibodies, they're not as good. They are cross-neutralizing, meaning you were gonna neutralize the virus to some degree, but they are not as powerful. And because they're not as powerful, and you still have lots of virus kicking around, that gave the virus opportunity to now start evolving against their substandard attack against it, so that new mutants could arrive, and again you have additional immune escape, and then you had another basically steric immune refocusing. So then the virus could keep evolving away and away, and that's exactly what we've been seeing. We keep seeing this wave after wave of new variants constantly coming out. Now, because the B cells that were originally triggered to start producing the original antibodies that used to neutralize the virus very well, because they were around, but when the B cells are supposed to now, when they trigger to produce new antibodies, they're supposed to be stimulated with a T cell that recognizes the same target. But because those B cells were no longer recognizing old targets, but new targets, they were not stimulated in the same fashion. And it took these B cells much longer time to mature, and this longer maturation process is why we also end up seeing with more increased switch towards IgG4 antibodies. So according to Dr. Bossche, IgG4 antibodies is actually a hallmark of the fact that that virus evolution took place towards immune escape, right? So, and it also is a hallmark of the fact that steric immune refocusing took place, and that's exactly what these other guys were seeing in their publication. So their publication, when the authors thought this is all good what they're seeing, it also could be supporting Dr. Bosche's theory that we're seeing evolution of the virus away from antibodies, which the virus now keeps escaping. Now, Dr. Bossche also believes that these IgG4 antibodies will also help trigger the evolution of the virus towards a more pathogenic state. And as a consequence, we're going to eventually end up with a virus that is going to be much more infectious, because that's what we have right now, but more pathogenic. And this is where we're going to have problems. And that's why he's unpopular. And to some degree, while I'm telling you this is science that I see that might be supporting his theory, the final conclusion of his theory, I'm really trying to find any science to disprove his theory. And I think maybe I found something. I'll make another video on that topic. But I have to wrap it up here because it's getting dark here right now. And this video took a little bit longer than I anticipated. But here it is, another paper that came on IgG4s by authors who also showed, and they were the first ones, as far as I know, who studied how vaccination impacts what's happening to the antibodies of these individuals. And these antibodies started to target different areas of the spike protein. And you could see where these spike protein, how the targets were being shifted away into totally different areas of the spike protein, totally far away from the receptor binding domain, which is the ideal target. So in a way, supports Dr. Bosche's theory, but I hope that his final prediction is still going to be wrong, that we're going to end up with more pathogenic virus. All right, that's all I have for you right now. I'm going to wrap it up. Thank you for all your support, and thanks for all the questions being submitted, and I look forward to seeing you in another installment on IgG4s, and I can already tell you there'll be more because there's more stuff that already came out since as well. Okay, bye everyone.